I'm Anna. I'm a PhD student out of the University of Kent and the Institute of Zoology at ZSL, and I work on the critically endangered wild camel in Mongolia. I'm fully funded by the Wild Camel Protection Foundation, and my genetic work has been supported by the NEOF lab at Sheffield. As an endangered species, there's not very much known about the camel, and my PhD aimed to answer some of the unknown questions about it. Both how many are left in its population in Mongolia, how healthy that population is in terms of genetic diversity, and how integressed that is with the domestic Bactrian camel. As an endangered species, there is also a captive breeding centre, and so we wanted to check if the diversity in the wild population is also represented in the captive population. The Wild Camel Protection Foundation has been working in Mongolia since 1997, and I'm lucky to be part of that continued project. My PhD started in 2019, but the fieldwork we began in 2018, and I've been working with the Wild Camel Protection Foundation since 2011. The nice thing about working on a long-term project is that I get to know the people in Mongolia and the colleagues that we work with out there, and I'm able to gain their trust and we're able to work together effectively. It also avoids helicopter conservation and going into the country and doing the work and disappearing again, and I think those relationships are very important. The lab work involved in my project was, first of all, to collect samples from Mongolia. We used non-invasive samples, so fecal samples, hair samples, and some tissue samples. We took those samples to the NEOF lab, extracted the DNA, then used microsatellite markers, sex-linked markers, and mitochondrial markers to determine diversity, integration, and sex of the animals that those samples came from. As well as collecting samples from the wild population, we also collected samples from the captive bred insurance population using tissue samples collected during veterinary procedures. We were able to then compare the diversity in the wild population with the captive population. The most interesting thing that I found so far was to determine that there is integration across the entire population in Mongolia. That's not across every individual, but across the extent of the area that the population covers. I was also able to determine that the wild population and the captive population share similar levels of genetic diversity and inbreeding, which is positive for the insurance population, as it means we've captured the correct level of diversity in that population. The future plans for the data collected in this project is to inform conservation management of the critically endangered wild camel in Mongolia, and in the end to save it from extinction. The first thing what we will do is to publish all these papers that we've written in both English and Mongolian, so that that information can be disseminated widely, and to get as much as possible to get it out there for people to learn more about the species. After that, the information is going to be used to produce a management plan for the species in Mongolia, and that will be given to the Mongolian government. NEOF has supported my project in terms of funding, lab facilities, and data analysis. I would recommend that others apply to NEOF to support their work. I've found both the help in the lab and their advice invaluable, as well as the funding meant that I could do much more for my project than I initially would have been able to do.